everyone. Good morning, afternoon, evening, depending on wherever you are in the world. Welcome to day three of the RWEsearch and Health Innovation Summit 2025. For the past 2 days, we have explored how RWE and health innovation is driving change and reshaping how healthcare is delivered in a bigger, better way across the world. Each session has shown us different perspectives around how a lot of advancements around science, data, artificial intelligence, and new business models are coming together to deliver this change. Today, we turn our attention to one of the most transformational areas that I personally have come across in last 1 year, which is around personalized cell therapy. In this session, we'll dive into the scientific foundations of how these therapies are done. We'll understand from Prudvi the current landscape of how they are being applied for some of the most complex and difficult to treat diseases, and what the future looks like in the next 1 or 2 years for us. To introduce the speaker, it's my pleasure to introduce Dr. Prudwidhar. He is the Medical Affairs Director at Miltenyi Biotec, India. Over the course of his career, he has worked at leadership roles at Takeda and Janssen, where he has been deeply involved in advancing strategic medical initiatives over the last 15 years. Dr. Prudvi is a physician by training and an ISB executive MBA, and he brings deep expertise in designing clinical trials, bringing innovative clinical therapies to life, and defining medical affairs strategies. I have personally had the privilege to interact with Dr. Prudvi over the last few months and understand about personalized cell therapies in detail. And I've been blown away by the transformation that is being shaped, which unfortunately is not well known in a lot of medical fraternity. It brings me back to this famous saying that the future is already here. It's just not evenly distributed. So I welcome Prudvi to take over and help us evenly distribute this future amongst the medical fraternity.
Thank you so much for that, Gaurav, and happy to do this at the conference. And hello, everyone. I didn't realize people are joining from different time zones. So, good afternoon, good evening, good morning from wherever you are joining. Before we go further, I am trying to figure out the slide controls. Okay, yeah, here we go. So today I'm going to talk to you about, while I'm calling it the fundamentals, present state, and the personalized cell therapy. The topic is very vast and very rapidly changing. What was true for, let's say, 2 years ago might not hold true now. There are so many advances, and the space is so dynamic that it's impossible to bucket all the cell therapies into one domain, because while we are talking about cell therapies, there are gene therapies that are evolving, and it's very complex, but we will simplify it and we will go further. So before we start the topic, I would like to declare my potential conflict of interest, because I'm a full-time employee at the Miltenyi Biotec. So there will be some of the content that will talk about how the cell therapies are being shaped by Miltenyi Biotec as well. So when we are talking about the cell therapies, let's take for an example autoimmune disease. So traditionally, there have been a lot of innovations that happened in this space, but mostly when we think about it, it was handled by typical immunosuppressions, right? Either your steroids or anti-inflammatory drugs and small molecules targeting. So the entire target was to reduce the inflammation in the space. Now what's happening is, as the space is evolving, now for the patients who still have that unmet need, there are new ways in which the solutions are being provided. One is, okay, if the regulatory part, since we know the immunity is like a balance, since the host immunity is attacking itself, so how can we improve the immune regulation? So we boost the regulatory part of immunity. So there are advances which are happening around the Tregs, and some of them are developing, let's say, CAR or T-Rex. There are advances in dendritic cells and natural killer cells, and some of this work is happening in India as we speak also. Now, while this part of the immune system is being taken care of, and some of the therapies are being advanced, uh, the typical way in which the physicians have already been trying to reset the system, whether it could be your cancer, whether it could be some of the autoimmune disease, is by going into the stem cell transplantation, right? So the auto-allo hematopoietic stem cell transplantation, it's nothing new, but the advances in the way it is being done is unprecedented right now. It's taking us to better and better levels because we can actually select the cells that we need to do the transplantation with. Now, the monoclonal antibodies have been there for quite some time. There are newer targets which are being investigated, and there are a lot of them that are coming up. And now, the newer space is the cell therapies. Some are targeting the B cells, some are targeting the T cells, and they are all being tried on various indications across the board. Now, before we dive further, when you think about which cell therapy should I even go for? Is it regulated? Is it unregulated? What's happening in this space? I hear the buzz. So let's keep it simple. The patient-specific therapy that we will, a physician should go for, or will even look for, or the industry partner will go for, is one that maximizes benefit and minimizes risk. So it depends on the indication and which cell to target to actually do that. Second modality in which this space is, advancing is one part of the system is, focused on targeting through the autologous sources. We take the source from the patient, we modify the cell, we'll give it to the patient now with enhanced cells so that, it can recognize either the cancer or will fix the problems in the immunity, and it is solved. Second part is allogenic source. It can be like we can produce in batches. We identify the right donor, we build that cell batches, and it can probably solve this problem at a scale. But why are these two processes diversified? Because one, the immune responses are very different. Autologous is very well tolerated. They are more safe for now. But allogenic are scalable. They are resource less intensive when you try to focus on it. So the therapy area is pursuing both, and the space is very dynamic as we see. And in order for the decision to happen, what are the forces that are changing the landscape? Is obviously the forces, pretty much the forces which shape most of the healthcare therapeutics. Here, the cost, of course, has to be taken into consideration, because at this point, the cell selection methodologies and with which it is being done, uh, is might not still be very affordable. We are glad for Indian innovation to reach at an affordable stage to the scale of population that we have, but the advances are still progressing to make it efficient. Now, when I mentioned the accessible part, if it's an autologous, obviously the process itself limits a bit of accessibility, because the patient will have to be available at the center of where the cell therapy can be produced, and allogenic can be more accessible. Effective, again, which pathway to go for, because sometimes one works against the other. The safety part, which we thought already we explained, and the need to develop a universal cell therapy, which can be like a prescriptive medicine that I give this for all the patient, it's likely to work. So these are all the forces which are shaping the current landscape that we are operating with. Now, having said that, while we will be inching deeper into the cell therapies, we will start with the basics. When we think about the transplantation traditionally, and this technology is not new, the idea is to focus on the immune resetting. We change what the problem area was, and we reset the immune system. So with that, I think the innovation was around both the autologous and allogenic stem cell transplantation. Then came that how, finding the donor can be very difficult. How do we advance into more and more precise way of doing this? Then came the ability to do the haploidentical, transplant, and this transformed the space. But when you are thinking about haploidentical transplant, it's really important to eliminate the graft versus host disease. So there are, technologies where you select the stem cells that are required. That is the CD34 selection. And then we do a bit of graft manipulation or graft engineering, and then further enhance this ability to do the stem cell transplant. So, like I mentioned, it's mostly to avoid the graft versus host uh, induction, because whenever we are giving the donor cell, they will recognize that, hey, it's not its own cell, and it will start attacking the host. And then to avoid this, there are various strategies that typically happen. I will start with the B and C first, because ATG is something that's followed in, let's say, China, that part of the world, and and there are technologies, let's say, post-transplant PTCY, and other immunosuppressants are given to actually make sure that some of these cells are taken care of to avoid the graft versus host reaction. But this is like a carpet bombing. How can we ensure that this happens in a very smooth way, and we actually select the cells that do not give the graft versus host reaction? Now, for example, when we are talking about the donor T cells, now the subsegment of the T cells are either alpha beta T cells or gamma delta. Now, gamma delta don't give graft versus host. If I need to select gamma delta T cells, there are technologies that allow me to pick up the gamma delta T cells and only infuse that into the patient. Correct. So this is what we mean by graft engineering, and the chart A illustrates how you can actually selectively deplete the cells which are a problem, and further enhance the transplantation criteria for haploidentical transplants, so that it's very successful. There is no carpet bombing, and this is very precise, and it enhances the patient's life opportunities a lot by a scale, right? So is this really hard to achieve? No, there are technologies which are very simple, and a lot of physicians currently are doing this in India as we speak. And there are technologies even supplied by the, let's say, Miltenyi, for example, which allow to do this. And this is the reality as we speak right now. Moving on to the next chapter, still on the topic of T cells. Now we know they are extremely important from the immune context, and they are the ones which recognize the cancer cells, the problematic antigens, and they attack these cells and further develop the cellular immunity. Now there was a phenomenon which is recognized, because the T cells were not precisely recognizing the problem elements in some of these cancer patients, and with that, uh, Carl June and many other scientists have recognized that we can actually modify the criteria with which the antigen can be selected. That is the origin of chimeric antigen T cell therapy, which we call the CAR T. There are various ways in which this is being done. The second, third, and fourth generation of CAR Ts are in the planning. So we are experimenting with a lot of co-stimulatory domain to ensure that it's the most efficacious and less toxic as a therapy. Is it only cancer that this CAR T is being used for? There is lot of research that's parallelly happening, uh, not just cancer, but also in autoimmune disease, which we will go a little bit deeper going further. This is being tried in fibrosis, and not just that, even there are some researchers trying in senescence, targeting senescence, and if you can, stop that progress. The CAR-T story cannot be complete without mentioning the story of Emily Whitehead. So she was the first living proof of the success of CAR-T. This was tried as an experimental, therapy for this patient who literally exhausted all the therapies that she was on, and there was no hope. And this was tried as an experiment, and we know the story. It's been more than a decade now, and Emily is the proof that this is a technology that's here to stay. And not just Emily, there are many patients across the globe who are getting benefited from this. And thanks to innovations in India, this is happening at, let's say, CMC Vellore. This is happening with ImmunoAct, Immunil. There are multiple players who are actually doing this now. So when we talk about the cancer and CAR-T role, it's kind of established that okay, this works. Now, this when this was tried in autoimmune, this was actually initiated by George Schett and team at the University of Erlangen, uh, where they actually tried to do this as a named patient program in their university, and tried doing it on the patients who were out of all the possible treatments. And this was the experiment because if we are targeting CD19, we eliminate the B cells, and how do we solve this? So just giving you example of one patient, not diving too much detail into the details of one particular trial, because that's not the overview of this topic. So one of the first patient that was treated with CD19 CAR-T was a young 20-year-old woman in the University Clinic of Erlangen. The patient presented with nephritis, pleuritis, endocarditis, arthritis, and all the classical symptoms of SLE. And the patient was tried on all possible regimens and completely refractory to including the novel treatments like belimumab. Now, this was the state. The complements were not doing well. Complement levels and the immunity, the B cells were depleted. They were higher than the range permissible. And as an experiment, the CAR-T was infused, and then the B cell depletion was logged. As you can see in this chart, within 7 days after CAR-T's infusion, there was complete depletion of B cells. And as this shows the expansion of CAR-T that is happening on the patient in the successive days. So the patient disease started drastically improving. The complement levels returned to normal. The proteinuria and all the patient symptomatic, complaints of the patient started improving drastically, and this happened within less than 44 days of time. And this was just one infusion of the CAR-T. And as you can see in this chart, it's not just the symptoms, but also all the signs of the patient and everything. Globally, it started improving. Within 44 days, the patient was completely symptom free, as you can see in this chart. The difference in this, so while the space is very evolving, right, there is still a lot of work being done. We don't know why some of these phenomena act different in oncology, why things happen different in autoimmune diseases. But we could note that the adaptive immunity recovers faster in, let's say, autoimmune, which is SLE, than your typical cancer cells. So this is being pursued further. So when we are talking about the B cell depletion, okay, so what happens? Do they reappear after the CAR-T infusion? Do we need a repeat of CAR-T infusion? What happens is, even when the abrogated B cells reappear after, let's say, close to 6 months, the current B cells after this happens, this you can mention that it's an immune reset, because the patients remain treatment free despite reoccurring of the B cells. And the B cells that are generated now are predominantly naive. So there is something that's happening that's causing a very deep reset. And now, as we speak, and to the current standard also, The patients are symptom free and they did not relapse so far, and the work shows that. And this was a picture captured at the University of Erlangen, where the patient is, disease free up to date 1,000 days after the first infusion, completely back to normal, and that means we cannot call her a patient anymore. She's a normal human being now, pursuing her ambitions, her life. That's the potential. That's the transformational potential of this therapy. How do we make this? Is it really simple? Okay, we hear about CAR-T. We do hear about the industry partners making this CAR-T. But how does this work? It's very precise. There are a lot of intermediary procedures which before we actually get to the product, because the patient's leukapheresis, if it's a sample, is washed, selected. You need to select the cells, you need to activate, you need to introduce the viral vector, transfuse with, and transduction happens, and you need to expand the cells, harvesting. There are many transfers and manual steps that typically happen in a typical process, and that's how it has been happening across the globe. But the technology is rapidly improving and innovating in this space. And some of the technologies, like let's say the Clinimax Prodigy is one of its kind. I don't know what happened. There is a bit of a zoom that happened. Okay. So, okay. So the closed technologies like Clinimax Prodigy and everything allow the manufacturing of the CAR-T in a very automated fashion, so that it's very simple. And this is the origin of the point of care, which produces the CAR-T at a hospital. So the CAR-T can now be produced in an automated fashion, and what we are working with majority of the institutes is the ability to produce these CAR-Ts at the institute itself. It's called the point of care CAR-T. Now, while the CAR-T cells are the current phenomenon and things are happening, there are a lot of other advances which are happening in this space, like the T-Rex, like I mentioned earlier. Now, this is the latest work that was presented in the Diabetologica very recently. So, the T-Rex, CD4 T-Rex are expressed and engineered, and then the polyclonal T-Rex are either modified with a TCR target or a CAR target, and their survival is enhanced a little bit with interleukin 2. And there is a proof that this is likely to work on the recent onset type 1 diabetes. This trial has shown that it is safe while the efficacy is still being proven. So there is some work that is happening in this space, and it's an exciting space to watch. Now, what's happening as we speak is also that some of these cell therapies are being tried where no other treatment is possible. Now, this is a single case example of a patient wherein T-Rex was infused to treat refractory ulcerative colitis and associating PSC. So the patients the technology is at the hospital. The clinicians themselves use the technologies to deplete the unwanted cells and to generate these regulatory cells and infuse. And this has caused, this has actually created this cell therapy when the cell therapy is infused, and this shows the improvement of ulcerative colitis and associated PIC just with a single infusion of autologous expanded polyclonal Tregs. So this is an exciting space to watch, and this is likely to grow. So what's happening in this space, if we are to speak in two diverse phenomena? One part of it is the technology with which we are advancing this. Another part is the needs of the biology. So let's talk about the technology first. So one part of the domain is to further automate these things and to make it simple so that there is a reduction in the cost of manufacturing. Second, to enable commercial readiness of this. So some of these are experimental therapies which are happening in early stage clinical trials or late stage clinical trials. How soon are these therapies ready to be produced at the hospitals at a commercial level? That's something that's happening. So that's what I mean by enabling the point of care manufacturing. That's around the autologous and off the shelf is something some of the industry partners are working on. That's the allogenic CAR T solutions. While this is all happening, there is some amazing work that's happening in the genetic engineering space, and there is further work happening on shortening the vein to vein time. That means uh, the patient's sample that's taken can be immediately sooner the we administer these cells. The chances that the recovery happens with the T cells and the persistence of the T cells and everything, the chance increases further. So there is this technology that's happening across the globe to make it uh, faster and faster to improve the manufacturing turnaround time. The cell activation and serum free expansion is a work that's happening, and some of the players are also working on enabling the scale at which this can be done uh, with the large volumes. And this is mostly happening in the allogenic domain. Now, in the cancer space, in the biology part of it. So, are we only targeting the hematological malignancies? No, there are a lot of work. There is a lot of work that's happening in the solid tumor targeting, and we are inching closer to success, including the recent one on full FOLR1 target that's happening on the ovarian and pancreatic cancer. There are CAR-Ts which are now coming up with dual antigen constructs, and some of them are triple antigen constructs, and there is it's a good amount of data that's being generated. Armored CAR Ts are happening wherein they can penetrate the solid cancer as well. Some of them will have the safety switches so that these novel CAR Ts do not turn into lymphomas, leukemias. And new cell types are being explored. CAR NKs are also being explored. Less differentiated phenotypes are being explored. Genetic engineering in the allogenic settings. And while we speak, because these are living cells, we need to improve the persistence and their ability to proliferate within the body. So that's been happening beyond the cancer. Also, like I mentioned, the autoimmunity and how we solve the graft versus host disease. And some of the early work is also happening on infectious diseases. So this is how the whole domain is actively progressing. And I'm personally excited to see where this takes us. This is a very dynamic space. A lot is happening, and much of this innovation is happening in India as well. I think this is going to transform the patient care as we go further. With that, I would like to conclude my presentation, and will look for questions. Okay,
Some of the questions at the bottom of the screen, which you can answer.
sure. So, Dr. Priya Nair mentions what are the indications where CAR T is used in India, oncology and immunology. How widely is it available and accepted? So, in India, the current approvals are mostly in DLBCL. There are the early stage trials that are happening in pediatric ALL. So that's where it's happening. And in autoimmune, let's say, you also mentioned immunology. There are early stage discussions that are happening to bring the technology from across the world to India to treat SLE. So I think we should see these approvals very soon. And any other questions? Okay, I am looking at the comments. So, what do you think are the key factors that will influence the adoption of personalized cell therapies in mainstream healthcare? I think one thing in India we cannot, you know, overstress is the affordability factor and how much it plays a role in influencing the decisions with which these therapies go, because the reimbursement here is a challenge, and it's the patient paying, and that is the single most factor which influence the adoption, because the technology part is already here, and a lot of this work is happening. That will actually influence the scale. Second is the awareness around it. While patient awareness is one thing, we do note that while the metros and some parts of it, they do realize that this is live now, the CAR Ts are available to treat the patients with, they can be managed. And second part is the awareness around it on cities beyond the metros. Third part is the apprehension around the cell therapies, because it's that fear of unknown, in my opinion, because it's very similar to how, let's say, monoclonal antibodies started their journey in India. So people were worried about the infusion related reactions. How do we do this and everything? And over time, right now, we know how efficiently the ecosystem right now handles it. So that's how I think all of this are shaping the personalized cell therapies in mainstream healthcare. Uh, the second question that I see is, given the high cost of cell therapy, how can we ensure that these treatments become accessible to broader patient population? I think one way this can actually happen is to by enhancing the point of care network. The hospitals being able to produce this at their own setup is the most efficient way, because there are technologies that are available to do that right now. This saves a lot of logistic time. The CAR Ts produced are very effective, very efficient, minimizes the turnaround time, minimizes the cold chain that is required to transport all that. So I think this is how all the hospital networks, and we can actually penetrate beyond the metros. And this, I think, is a single most lever which can actually enhance the access to a broader patient population, right? So then I see another question. What are the biggest technical or logistical challenges in scaling up personalized cell therapies for widespread clinical use? I think technical is open versus closed debate. There are open manufacturing platforms, there are closed and automated platforms. The open platforms come with its own exchange of the process, like I mentioned. That is one of the challenge, and the closed is actually a very simple, requires less requirement for the clean room, and that's the best way. And the logistic challenges are also minimalized when we adopt to the closed and automated platforms, because everything happens at the hospital network, and we don't really have to invest in the logistic challenges. One more question is, how are the regulatory bodies like the FDA approaching the approval process for personalized cell therapies, and what hurdles remain? So the regulators are, traveling along with the industry partners and innovators on this. Most of the work is developed by the academic institutes, right? And they are all working along with FDA, US FDA, and not just US FDA, but European agencies, and the same with India. So a lot of discussions are happening. I think they are very open, because the regulators also see the transformative potential of these therapies, and they are working uh, together in a sandbox environment to make this more effective and to clear the hurdles on this, to make it available to larger and larger population. So, where do we currently stand on terms of clinical trials for personalized cell therapies? What have been the most promising results so far? I think when we are talking about personalized cell therapies, there are so many trials that are happening in the global stage. And if we are talking about India's context, some of these therapies have even promised overall response rate of about 92 to 96% also. So it's very promising. Uh, though they are early therapies, the patients are relapse free. Most of them respond when they are left with no other therapy possible, and we see a lot of things moving there. I think the scope is enormous, and also in the space of graft engineering, like I mentioned, the survival impact is a lot because transplants are already transformational for the survival, and the ability to select and engineer the transplants is further promising. So I think these are the questions. Are there new? So there has been a positive data on the use of CAR-T in scleroderma, yes, but beyond SLE and scleroderma, how do you see its potential? I think it is being still tried in a lot of other autoimmune spectrum also beyond scleroderma. There is some early work that's happening in myositis. There is uh, some work happening in the space of rheumatoid arthritis also. So while the results are, I would say it's more of uh, let's say 3 patients, 5 patients, which typically happen in a basket trials like in phase 1. This is being expanded right now. Lot of early work is happening, but it's showing promising results already. Uh, like it's mentioned, beyond SLE and scleroderma, myositis, it's showing positive results, and rheumatoid arthritis, it's too early to comment on that space.
It's been a enlightening session for all of us. Thank you so much.